Next one is multiple fission. So far we have seen only about binary fission. The next one is multiple fission. Multiple fission is, we have seen in binary fission one organism splitting into two. Multiple fission is the nucleus splits into many nuclei, and later what happens is, as much as how much nuclei is present, the same part cytoplasm also divides. Then this cytoplasm encircles the nuclei, and later each of that will develop into a new organism. This is called as multiple fission. This is seen in Vorticella and Plasmodium. So in Vorticella, if you see, it divides into many cells. As long as the process gets completed, the individual remains in the parent body itself. It does not separate until the process is complete. That is why it is called as repeated fission. Next one, if you see Plasmodium. Plasmodium, already we have seen life cycle of Plasmodium in 10th standard, and you have read that it has two life cycle. One is in the human being, the other one is in the mosquito. In human being, we have read about the zyzone stage. So if the multiple fission takes place at the zyzone stage, that process is called as zygogony, and the young ones which are formed are called as merozoites. Next, oocyte stage, it is in the mosquito. So when the Plasmodium life cycle continues in the mosquito, that process, the multiple fission process that takes place inside the body of the mosquito, that is what is called as oogony. And the young ones which are developing from that process is called as sporozoites. So you can see in this diagram, the zygone, that is the nucleus, it breaks into many nuclei, and then the cytoplasm splits and it encircles the nuclei, and later the zygone wall ruptures and the young ones are liberated out. So this is a type of multiple fission. Next one is encysment. As I told before, during the introduction, I told you encysment occurs during unfavorable condition. That is, when the condition is not suitable for the organism to survive, then it encases around itself to protect itself from the unfavorable condition. So that is what is called as encysment. Encysment usually takes place like this in amoeba. That is, 3-layered cyst wall will be formed, whereas you have seen in amoeba it has an irregular shape. Pseudopodia will be seen. This pseudopodia will be withdrawn and 3-layered cyst wall will be seen covering it. Then the nucleus divides and it forms the cytoplasm forms encircles around it. That is what is called as pseudopodiospores. And when the condition becomes favorable, the cyst opens or bursts or ruptures, and then the young amoebae are released out. So this is one method and it is called as encysment. So this occurs during unfavorable condition. So 3 protective layered cyst wall will be formed around the amoeba, where the pseudopodia structures will be withdrawn or that will be lost, and it encircles like a ball-like structure. So the nuclei splits as well as the cytoplasm splits and encircles, and when the favorable condition returns, the ruptured cyst wall will liberate the young amoebae. Next one is strobulation. Strobulation, in this process we have read about transverse fission. The same like that, but many transverse fission occurs, then it is called as strobulation. Example for this is Aurelia. So you can see in this is young strobila. So here you can see in Aurelia, this is an young one. The next one is the matured one. So you can see each one, when it matures, it can develop into a individual organism. So the transverse fission occurs in each strobila. So they can survive individually. So this type of fission is called as strobulation. Next one is plasmotomy. So plasmotomy, it's a special type of fission in which a multinucleate protozoan divides into 2 or more multinucleate daughter individuals. So far what we have seen is, all those organisms what we have seen so far, they possess only one nucleus. That nucleus splits into or divides into many nuclei. But here in plasmotomy, if you see, already the parent possesses multinucleus, that is many nucleus. This multinucleated parent or the protozoan will divide into 2 or more multinucleate daughter individuals. So we cannot say that both the daughter possess same number of nucleus, but later they get that constant number of nucleus by the nuclear division. So example for this is Peleomyxa and Opalina or opalinates. So this is an example for plasmotomy. Next one is sporulation. Amoeba multiplies without encysment. So already we have seen a process with encysment. Without encysment, it is called as sporulation. So you can see the pseudopodia structure is as such. The pseudopodia is not withdrawn. The same pseudopodia structures will be seen. Only thing is the nucleus splits into many fragments. Each of these fragments are called as chromatin blocks. So the nucleus splits into many fragments. Those fragments are called as chromatin blocks. These chromatin blocks are encircled by cytoplasm, and it is completely covered by a spore cyst. So this spore cyst or spore case encircles each of the nucleus when the condition becomes favorable. And again, this sporulation also takes place only during the unfavorable condition. When the condition becomes favorable, again the spores will be ruptured and it releases the amoeba. So this is one type of reproduction. Next one is budding. Budding is a very easiest type of reproduction, asexual reproduction. Budding is a very easy type of reproduction. So budding is of two types. One is exogenous budding, another one is called as endogenous budding. First we will see what is exogenous budding. Exogenous budding, buds, if it is formed on the outer surface of the parent body, then it is called as exogenous budding. Usually when this exogenous budding takes place is when the condition is favorable for the organism, or when the amount of food it receives is in plenty. The ectodermal cells and the endodermal cells together, it evaginates. So it forms or protrudes outside the body. Then slowly it develops tentacles like the parent body, and when it gets matured, it detaches from the parent body and develops into a separate individual organism. So this occurs usually during the favorable condition, or when the hydra receives plenty of food. So this is one method. This is called as exogenous budding. Exogenous budding, where the buds will be formed outside the body of the parent. Next one is endogenous budding. So here buds are formed inside the cytoplasm of the organism. Example, Noctiluca. So cytoplasm of the cytoplasm develops into bud-like structure. We call it as internal buds. So that is what is called as endogenous budding. Next one is called as Jamul formation. Jamul formation is more or less similar to endogenous budding. So here if you see, food-laden archaeocytes are formed. So each of these nucleus-like structures which are seen in the center, they are all food-laden. Food-laden means completely filled with food materials. So we call that cells as archaeocytes. So usually this type of Jamul formation is seen in freshwater and some marine sponges. So here what happens when the condition, this is also during the unfavorable condition, when the condition becomes favorable, the sponge disintegrates and the Jamules will be hatched out into new sponges. So this is one type of asexual reproduction, that is Jamul formation. Next one is fragmentation. Fragmentation, that is the parent body breaks into fragments and each fragment develops into a new organism. So this is well seen in sea anemones. So if you see fragments, you can say again cutting into two halves or like that, similar to fission. But so we should know what is the difference between fission and fragmentation. So here fission, it is defined as the process in which a unicellular organism splits into two or more daughter cells. So that is the main difference. Fission occurs in unicellular organism and where it gives rise to two or more daughter cells. But fragmentation, it is a breakdown of a body. Their cells, here body, and it occurs in multicellular organism into pieces which then grows and forms complete organism. So here gives rise to two or more daughter cells, where fragmentation, if you see, it is a breakdown of body in the multicellular organism into pieces, and that develops into a complete organism. This is unicellular, that is in multicellular. So example we have seen already, amoeba, paramecium, leishmania. Here it is Spirogyra and sea anemones. So in this picture, if you see, this is a picture of sea anemone. So this is the lobe. You can see splitting or a fission in the lobe. So this splits into two, and that each develops a pedal disc, and above it develops the tentacle-like structures and separate from the parent body and remains as a separate individual. Next one is called as apolysis. Apolysis actually is not a method of reproduction, but it is a process where regular cut-off of gravid proglottids occur. What is called gravid proglottids is, proglottid means each segment of the tapeworm is called as proglottid. Gravid proglottid means ripe or mature proglottid is called as gravid proglottid. So gravid proglottids will be seen in the posterior end of the tapeworm. What is the need? What happens during why it is regularly cut off? So what is the reason? What is the importance of that is to transfer the developed embryos from the primary host to the secondary host. That is, each of that contains the embryos. These embryos has to undergo life cycle in two organisms. One is the primary host man and the second one is the secondary host pig. So in order to complete its life cycle, the embryos has to be released from the parent body. For that purpose, apolysis takes place. Next one is regeneration. Regeneration is regrowth in the injured region. So this process of regeneration was first studied by Abraham Tremblay in the year 1740 in Hydra. So in regeneration we have two types. One is called as morphallaxis and the next one is epimorphosis. Morphallaxis, epimorphosis. Morphallaxis, where the whole body grows from a small fragment. Example, Hydra planaria. So morphallaxis is a process where the entire body will be formed from a small fragment. That is what is called as morphallaxis. But epimorphosis is replacement of lost body parts. That is what is called as epimorphosis. So when the whole body grows, we call it as morphallaxis. When only a part of that grows, last part grows again, then it is called as epimorphosis. This epimorphosis is of two types. One is reparative, another one is called as restorative. Reparative means regeneration of only certain damaged tissue. For example, any part of the body gets damaged. Again, when it gets healed, that type of mechanism is called as reparative. Next one is restorative. Severed body parts develop again. That is, if it is cut off, any part of that is cut off, and again if it develops, then it is called as restorative. So you can see in this diagram. So here this is the first one, regeneration in Hydra. So whatever is given in black is old part. So from the old part, the new part has been developed. The next one is reparative regeneration. Here if you see the tissue, a little part of the tissue alone is damaged, that is again regenerated. So reparative regeneration in starfish. So you can see the old part regains its last part. Here the tissue which has been lost is developed. So this is another type of regeneration. So reparative, restorative. So thank you children. This is what is about asexual reproduction.